Dear students, the topic is genetic code.
Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment.
Dear students, as DNA molecule is a genetic material, it carries genetic information from cell to cell and generation to generation. And a DNA molecule is composed of three kinds of moieties. You know which are they? Phosphoric acid, deoxyribose sugar and nitrogen bases. Among these three, the nitrogen bases vary from one segment of DNA to another. So, the information might depend on their sequences. Molecular geneticists try to understand these coded language for how these genetic information transmit from one generation to next generation, and they identified there is a coded language for this, and that is called genetic code. We can discuss in detail about genetic code, its characteristic features and significance in detail from the following slides. You know, the central dogma process in molecular biology is DNA undergo transcription and form RNA, then RNA translated into protein. So transcription and translation are the two processes of central dogma. The nucleotides adenine, guanine, thymine and cytosine found in DNA are organized into codons. The collection of codons is called genetic code. For 20 amino acids, there should be 20 codons. Each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. So, nucleotide consists of 4 combinations and nucleotide consists of 16 combinations and nucleotides of 64 combination. So, these are most suited for 20 amino acids. See here, this is the standard genetic code. First letter, these 4. First letter, then second letter, third letter. These 3 join and form a codon. Next we can see the characteristics of genetic code. The first one is specificity. Genetic code is specific or unambiguous. A specific codon always codes for the same amino acid. Example, UUU codes for phenylalanine. It cannot code for any other amino acid. Second character is universality. That means genetic code is universal. In all living organisms, genetic code is the same. The exception to universality is found in mitochondrial codons where AUA code for methionine and UGA for tryptophan instead of isoleucine, and termination codon respectively of cytoplasmic protein synthesizing machinery. AGA and AGG code for arginine in cytoplasm, but in mitochondria they are termination codons. Genetic code is redundant, also called degenerate. Although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons. Except tryptophan and methionine, each amino acid has multiple codons. That is shown in the table. Next is genetic code is non-overlapping or non-punctuated. All codon are independent sets of 3 bases. There is no overlapping. Codon is read from a fixed starting point as a continuous sequence of bases taken 3 at a time. The starting point is extremely important and this is called reading frame. Next is nonsense codon or stop codons. There are 3 codons out of 64 in genetic code which do not code for any amino acid. These are called termination codons or stop codon or nonsense codons. The stop codons are UAA, UAG and UGA. They encode no amino acid. The ribosome passes and falls off the mRNA. Initiation codon. AUG stands for methionine, is the initiation codon in majority of protein. In a few cases, GUG may be the initiation codon. Methionine is the only amino acid specified by just one codon, that is AUG. Next is Wobble's hypothesis. The rules of base pairing are relaxed at the third position so that a base can pair with more than one complementary base. Some tRNA anticodons have inosine at the third position. Inosine can pair with uracil, cytosine or adenine. This means that we don't need 61 different tRNA molecules, only half as many are required. First two bases in codon in mRNA, 5' to 3' base pair traditionally with the second and third base of the anticodon in tRNA. That is also 5' to 3'. Non-traditional base pairing is observed between third base of the codon and the first base of the anticodon. The reduced specificity between the third base of the codon and the complementary nucleotide in anticodon is responsible for wobbling. Significance of genetic code. Mutation can be well explained using the genetic code, like point mutation, silent, missense and nonsense mutations, then frame shift mutations.
Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.